Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Jenny England, staff scientist from Thermo Fisher Scientific.
Thank you for the introduction, Alex. As you mentioned, my name is Jenny England, and I work at Thermo Fisher in the Poros Purification group. And today we'll be talking about enabling mRNA-based therapeutics development through efficient and scalable mRNA purification methods. So just a quick agenda. I'll start with an intro into mRNA therapeutics, a discussion on the current chromatography methods to purify mRNA, our Poros oligo-DT 25 affinity resin, which we offer as a solution for mRNA purification, an overview on affinity capture development and resin reusability, and lastly, an elution optimization study that was recently completed by my group. So synthetic mRNA has many diverse applications in the treatment of cancer disease. For example, in the clinical pipeline, mRNA is used for cancer immunotherapy, vaccines against rare and common diseases, allergy tolerization, protein replacement and production, gene editing by using TALENs or CRISPRs to correct defective genes, and genetic reprogramming for stem cell induction. Additionally, methods for mRNA therapy include direct injection, ex vivo injection of transfected cells, and transfection of genome editing enzymes. Therefore, these diverse applications and methods for injections will require a robust and easy-to-use purification platform to drive the success of all of these mRNA applications. This slide shows how nucleic acid therapeutics have become an emerging technology, and the raw material needs and regulatory requirements are still evolving and not yet standardized in the industry. So for example, therapeutics typically follow 4 stages. There is the nascent phase, where technology shows high innovation and potential, but have yet to demonstrate value. The growth stage, where the impact begins to crystallize as the technology becomes more defined and measurable. And the emerging state, where the vector is established clearly by diffuse metrics such as scientific literature, patents, VC activity, and grant focus. And lastly, the mature phase, where the technology is mature and knowledge is well established. RNA therapeutics are expected to follow a similar curve to mAbs and viral vector gene therapy. And the COVID-19 pandemic has moved mRNA quickly past the nascent and growth phase into the emerging modality phase. However, in order to reach the mature technology area, there needs to be considerable investments made into optimizing the production of mRNA. mRNA therapeutics has progressed significantly over the 30 years from early testing to clinical use. The first proposal of mRNA vaccines began in 1990 with a successful study performed in mice. This was followed by the first vaccination with mRNA encoding a modified cancer antigen, which was observed in 1995 with the first clinical trials with ex vivo DCs transfected with mRNA against cancer in 2002. In 2005, an important milestone was achieved for RNA therapeutics in the demonstration that nucleoside-modified RNA is non-immunogenic. More progress was made for methods of delivery of mRNA in 2009 and 2010, and this was followed by a flu and RSV-specific mRNA vaccine preclinical study in 2012. It wasn't until 5 years later that the first use of a personalized mRNA vaccine was tested in 2017. And of course, the acceleration of mRNA therapeutics was ignited by the global need for a vaccine against COVID-19 in 2020, with the first approval of the mRNA COVID vaccine in 2021. Despite the growing application of mRNA therapeutics, many challenges still remain in mRNA purification. Current chromatography methods of RNA purification include reverse phase, ion exchange chromatography, size exclusion chromatography, hydrophobic interaction chromatography, and affinity chromatography. Reverse phase chromatography has high resolution but has limited column capacity, and the use of expensive, flammable, and toxic chemicals make it not ideal for large-scale manufacturing. Ion exchange and hydrophobic interaction chromatography enable for scalable solutions, but ion exchange can have limited column capacity and recovery. May need toxic chemicals, and purified product can contain traces of these elution salts. While HIC can serve as a replacement for reverse phase, it is non-selective. The last chromatography method, affinity chromatography, can be used as a scalable platform solution for mRNA purification. Affinity chromatography is selective to the poly A tail. Native purification is possible and can serve as a platform solution for a wide range of mRNA sizes. However, disadvantages include an additional polish step to remove product-related impurities. This slide summarizes the mRNA therapeutic workflow from DNA template preparation, mRNA synthesis, and downstream purification. Our porous products can be used at both the plasmid and mRNA purification stage. For mRNA purification, we offer the porous oligo-DT affinity resin and our IEX and HIC chromatography resins for the polish step. After the IVT reaction, the full-length mRNA is selectively captured by the poly A tail with our porous oligo-DT affinity resin. Because of the specificity to the poly A tail, only the full-length mRNA is remaining after the affinity capture step. Additionally, this step enables the removal of process-related impurities such as DNA template, nucleotides, enzymes, and buffer components. An additional polish step can also be done to remove double-stranded RNA and uncapped RNA from the final product. This step can also remove secondary RNA structures if needed. The porous oligo-DT resin has been specifically designed for the purification and isolation of mRNA from the IVT reaction. The mRNA is captured through AT base pair hybridization, which is illustrated on the right-hand side of the slide. A DT25 poly-T ligand with a proprietary linker is attached to a 50-micron porose bead. The resin has a dynamic binding capacity of up to 5 mg per ml for a 4,000 nucleotide of mRNA, with a greater than 90% recovery, and it also has excellent scalability and is non-animal derived. As I mentioned in the previous slide, our ODT resin is on our unique porose bead, which has 3 main attributes that differentiate from other chromatography resins. The first is the bead is made out of a polystyrene divinyl benzene material that will result in stable column beds with linear and scalable pressure. This enables the operation of a high linear flow rate with modest pressure drops. For example, the operation at 800 centimeters per hour in a 20 centimeter length column will result in a 2-bar pressure drop across the column. This is a small change in comparison to a classic soft gel resin that shows a typical exponential increase in back pressure as linear velocity increases. Additionally, due to the polymeric nature of the backbone and the robust covalent chemistry to the beads, the porous beads have a very robust physical and chemical stability and are chemically stable to pH 1 through 14, high salt concentrations, and detergents and denaturints. This enables the use of aggressive cleaning solutions, which will improve resin lifetime and reuse. The second attribute is a large pore structure that results in reduced mass transfer resistance in comparison to other resins. This large pore structure enables linear velocity increases with very little loss in capacity and resolution and improved process productivity. And finally, The third attribute is a 50-micron average particle size. The small particle allows for less band bonding in packed beds and therefore improves the resolution of molecule separation and improved impurity removal. Now that I've given you an overview of our porous oligo-DT product, I will start and discuss an overview of the affinity capture development. I'll walk through understanding mRNA stability, binding capacity experiments, and purity and yield analysis. One thing that I wanted to point out today is that the oligo-DT resin can easily be scaled from small scale to large scale. Conditions can be optimized in a 96-well plate or the robocolumns for your specific mRNA construct to maximize mRNA purification. Once the ideal conditions are determined, they can then be verified with our pre-packed columns on the bench scale. These conditions established at the small scale can be applied when scaling up your mRNA purification to the liter scale. As previously mentioned, it is important to optimize your mRNA process for your specific molecule. This slide shows the precipitation of mRNA as a function of increasing salt concentration. The A600 measurement was used to determine the precipitation point of mRNA. Two different salts were also tested, sodium chloride and potassium chloride. As you can see in the graph on the left, in the presence of 1.4 molar sodium chloride, precipitation of a 2,000 nucleotide mRNA is observed. For a 1,000 and 3,000 nucleotide, mRNA precipitation is observed at 1.8 molar sodium chloride. On the other hand, for potassium chloride, precipitation is observed for a 1,000 nucleotide mRNA at 1.8 molar salt and 2 molar potassium chloride for 2,000 and 3,000 nucleotide mRNA. In summary, mRNA precipitation is dependent on the construct size, sequence, and type of salt used. Another important factor is to determine how the mRNA load concentration and residence time can impact the binding capacity. For a 3,000 nucleotide mRNA molecule, we can see that an increase in the sample load concentration from 0.1 mg per ml to 0.5 mg per ml results in an increase in the 5% dynamic binding capacity, while a lower concentration actually results in a decrease in the 5% DBC. Additionally, the slower the residence time, the better the binding capacity, where the best binding capacity was observed at 0.5 mg per ml sample load with a 4-minute residence time. This slide summarizes the impact of mRNA size on binding capacity and recovery. For a 1,000 mRNA, a binding capacity of 4.5 mg per ml is observed at 5% breakthrough. However, a 4 mg per ml of resin binding capacity is observed at 10% breakthrough for a 2,000 nucleotide size mRNA. For a 3,000 nucleotide mRNA molecule, you could obtain a 3 mg per ml resin binding capacity at 10% breakthrough. These binding capacities were all determined at a 2-minute residence time and a 97 centimeter per hour column load. Additionally, a greater than 90% recovery is observed for all 3 mRNA sizes. An example chromatogram is shown for the purification of a 2,000 nucleotide mRNA from an IVT mixture with our porous oligo-DT resin. The sample was loaded at 2 mg per ml of resin load with a final sample concentration of 0.2 mg per ml, which was diluted in 10 millimolar tris, 0.8 molar sodium chloride, 1 millimolar EDTA at pH 7.4 binding buffer. During the sample loading, only the mRNA with the poly A tail will bind to the ODT resin, and the DNA, enzymes, and nucleotides will flow through the column. After sample loading, the column was cleaned with the 2 washes, equilibration buffer, and a low salt wash at 0.1 molar sodium chloride. The mRNA was eluted with RNase-free water, and the column was cleaned with 0.1 molar sodium hydroxide. The recovery and impurity removal is shown on the slide. in which the mRNA recovery for 2 mg per ml load is shown to be greater than 90%, and the enzyme impurity removal in comparison to the load and the flow-through is minimal, below the detection limit in the elution fraction. So therefore, a high recovery and purity can be achieved with our porous oligo-DT independent of sample type. Along with the greater than 90% recovery, the ODT resin can be reused multiple times without loss of recovery over 10 cycles. This slide shows the elution peak over 10 cycles for the purification of an IVT mixture. As you can see, the traces overlay identical to the initial run, and no carryover is observed from the previous purification. Additionally, a 90% recovery is observed for the 10 cycles. In addition to the recoveries and the impurity removals, the cleaning and stability of the ODT affinity resin can be reused over a large number of buffers. This graph summarizes this cleaning for the ODT resin for 0.1 normal hydrochloric acid, 0.1 normal sodium hydroxide, and 0.5 normal sodium hydroxide for incubation time over a 48-hour period. The ionic capacity is measured on the y-axis and is a measure of the ligand density on the resin. Looking at the plot as a function of time, you can see that the ionic capacity stays above 90%, independent of the buffer for the 48 hours. So therefore, our ODT resin demonstrates good stability over a wide range of pH conditions, 1 through 13, and can withstand up to 0.5 normal sodium hydroxide, allowing for stringent cleaning and sanitization. Lastly, I'm going to be talking about our elution optimization study that has been done. What I'll present is high throughput screening of different elution conditions and comparing their respective recoveries. After we identified the top elution condition, we moved into 1 ml column experiments, implementing those elution buffers with recovery comparable to water. A comparison of citrate-only elution buffers and their normalized % recoveries in comparison to RNase-free water is shown in the graph. Lower recoveries were observed for citrate buffer at pH 5, independent of the ionic strength or presence of EDTA. Comparable recoveries to water were observed for citrate at pH 6, with similar recoveries for 1 millimolar and 5 millimolar citrate. Interestingly, the addition of EDTA to 5 millimolar citrate pH 6 resulted in a decrease in the recovery by 25%. The better elution recovery of citrate at pH 6 could be explained by the charge of the citrate molecule. Citrate has a pKa value of approximately 6, so at pH 6, one of the carboxylic acid groups could be deprotonated, resulting in less protons on the molecule and less hydrogen bonding capabilities. This could have a destructive effect to the AT base pair interactions and result in better recoveries from the ODT resin. This graph summarizes the TRISH-only elution buffers. TRISH buffer at pH 7 did not see a large difference in recovery for 5 and 10 millimolar tris, but a large decrease in recovery was observed for 25 millimolar tris. While at pH 8.0, 5 millimolar tris resulted in increased recovery, and an increase in the ionic strength from 5 to 10 millimolar resulted in a 50% loss in recovery, and again, a poor recovery was observed for 25 millimolar tris at pH 8. After completing the high throughput screening experiments, we then scaled this up to 1 ml column verification runs. So we can see here a summary of a graph that shows recoveries for a 1,000 nucleotide and 2,500 nucleotide mRNA constructs, and we compared this using water, 1 millimolar citrate pH 6, 1 millimolar citrate, 1 millimolar EDTA pH 6, and 5 millimolar tris, 1 millimolar ETA pH 8. We used our porous T pre-packed column, and we loaded a purified mRNA, which was generated by a PCR template. We did a resin load of 2 mg per ml of resin, and the mRNA concentration in the load was 0.2 mg per ml. The column run method is shown in the table on the bottom right. The EQ buffer was the same buffer that was used for the high throughput screening, and we did a 5-minute residence time for the load. This is followed by an EQ buffer wash, and then the low salt wash was the 0.1 molar sodium chloride, and then the dilution buffer was varied. And then we followed by a sip with the 0.1 sodium hydroxide, and then we re-equilibrated with the EQ buffer. So going back to the graph, we can see that if water has 100% recovery, citrate and citrate with EDTA had approximately a 90% recovery for the 1,000, the 2,500. A 90% recovery was observed for the tris EDTA buffer for the 1,000 nucleotide, and we did not test for the 2,500 nucleotide. So I'd like to take this time to say thank you for listening to the presentation, and I'd like to acknowledge my team who was responsible for executing all of this work today, as well as others who have helped with generating the experimental design and analyzing the data, and I'm happy to take any further questions. Thank you.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, what purification method would be recommended for dsRNA removal?
So the current method for dsRNA is the use of reverse phase HPLC. However, as I mentioned, for HPLC, the use of organic solvents as well as high temperatures for this method does not make it ideal for scale-up. Therefore, we would recommend to purify double-stranded RNA the use of our anion exchange resins. We have our D50s as well as our HQ and XQ resin, or our hydrophobic interaction chromatography resin. There's the benzyl ultra resin that I would recommend for it. And so, yes, so as I said, we offer the anion exchangers and the HIC resins, which can be used for double-stranded RNA removal.
And what are some recommended conditions and additives to screen to optimize binding of my mRNA molecule to the ODT resin?
So I touched base a little bit about this in the presentation. We use different salts and concentrations can be used to optimize binding. However, you can also test different buffers such as tris and potassium phosphate. You can also try different additives such as EDTA and magnesium chloride. But as we showed in the presentation, just testing the different salt concentrations with either sodium chloride or potassium chloride, it's really important to understand that salt precipitation point when trying to optimize binding to the ODT resin.
How would the buffer, additives, and residence times change for larger mRNA molecules such as self-amplifying mRNA?
Yeah, so for larger mRNA molecules, such as self-amplifying mRNA or anything greater than, the traditional COVID vaccines, it's really important to understand that ideal salt concentration for binding to the ODT resin. So doing a salt screening study would first be recommended. We would also recommend those lower sample feed concentrations, so about the 0.2 mg per ml sample concentration load, and we would recommend a longer residence time of about 5 to 8 minutes for dealing with larger mRNA molecules.
All right, so it looks like we're running short on time. If we don't get to your question, we'll be passing them along to Jenny, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, can you elaborate on the experimental details for the high throughput screening experiments for the alternative elution studies?
Definitely. So for the high throughput experiments, we used 20 microliters of resin for each condition, and we did those conditions in duplicate. The resin was equilibrated with the same EQ buffer that I mentioned in the presentation. It's at 10 millimolar tris, 1 millimolar EDTA, 0.8 molar sodium chloride, pH 7.4. So we would equilibrate the resin with 200 microliters of the EQ buffer 3 times. We did this with our TCAN system and then centrifuge to remove the added EQ buffer. And before loading the mRNA onto the resin, we would take the sample and do a dilution into 200 microliters, total sample volume. We would take that, bind it to the resin, incubate it for 1 hour while shaking at room temperature, and then we would wash with the EQ buffer and the low salt buffer 2 times with the same 200 microliters. And then we would elute the sample with adding the elution buffer, and we would incubate that for 5 minutes. And we would do that 2 times. And then we would finally strip with the 0.1 molar sodium hydroxide.
Okay, great. Well, thank you, Jenny, and thanks to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.